Hello everyone and welcome to my channel. This is Pooja. Now, have you ever found an unknown DNA sequence and wanted to identify which organism it belongs to? Or maybe you wanted to compare your sequence with millions of sequences available worldwide. That is exactly where the BLAST becomes extremely powerful. Yes, the today's video is all about the BLAST, which is in the series of our NCBI. Okay. In this video, I will show you step by step how to use NCBI BLAST to sequence identification and analysis. First, let's see what is BLAST. So it stands for Basic Local Alignment Search Tool, and it is one of the most widely used bioinformatic tools in the worldwide. It compares your DNA or protein sequence with sequences present in biological databases and finds similar matches. There are lots of uses of BLAST. It is used for sequence identification, species identification, homology search. If you want to identify any functional gene like AMR gene detection, or if you want to compare your organism with other organisms, so for comparative genomics you can use the BLAST, then mutation analysis, and if you want to confirm any gene like disease gene, so for that also you can use the BLAST. There are various types of BLAST. On the basis of NCBI provide different types of BLAST tools. So BLASTn is used for nucleotide versus nucleotide comparison. We will see that step by step. So first I will show you. Go to the NCBI website. From here you can choose BLAST. Okay. Now you can see here the types of BLAST. So first one is BLASTn. Okay, if you can click here. Now let's see here. You can see here BLASTn. So it is used for nucleotide versus nucleotide comparison. Then BLASTp. It is used for protein versus protein comparison. Then BLASTx. It is used or translate nucleotide sequences into protein and compares them with protein databases. The next one is the tBLASTn. So it compares the protein queries against translated nucleotide databases. And the next one it is tBLASTx. So it compares translated nucleotide sequences against translated nucleotide databases. So on the basis of which you want which is your query, you can use any one of these. So now let's perform a BLAST search. So here you will see different BLAST options. So for this tutorial, select BLASTn, which is already selected because we are comparing nucleotide sequences. Now I am going to paste here. Here you can see enter query sequence. You can add here accession number or FASTA sequences. I have already given the tutorial of FASTA, how to download the FASTA sequence, and in that FASTA sequence, itself it started with the accession number. I will show you. So this is the FASTA sequence of Metabacillus malachi, and you can see here the first it is started with accession number, which is universal. You can copy from here, Control C, and you can paste simply it in the box. Okay, I have pasted the accession number. Now here in the field job title you can see it is given Metabacillus malachi strep ppm 16S. Okay. Now from here we have selected the program to optimize highly similar sequences. So we are going to select on BLAST. Okay. So after selecting the database, click on after clicking the BLAST button, NCBI, now you will compare your sequence against millions of sequences in the databases. You can see here. Now how we can interpret the results. Now let's understand the BLAST result. The first important parameter, it is query coverage. So query coverage match with the query coverage tells us how much of our sequence match with the database sequences. Next is the percent identity. You can see here percent identity. So higher the percentage means greater similarity between the sequences. And the another very important parameter, it is E value. So the E value represents the probability that the match occurred by chance. So lower the E value means higher significance and more reliable match. So 0 E value, it is more reliable. You will also see alignment score from here. Okay, so how much alignment is there? So from 1, 3, 5, 6, 734 bases are aligned. So from this organism it is different. Okay, so that's why BLAST, it is extremely important in gene identification, mutation analysis, AMR gene confirmation. So this is how you can use NCBI BLAST to identify and analyze biological sequences. And if you found this tutorial useful, don't forget to click the like the video, subscribe to the channel, and for more NCBI and bioinformatics tutorials, press the bell icon. Thank you for watching.